There are about 100 trillion cells in your body, but you started out as just one. How can one cell turn into a million? Where do cells come from? Just like all organisms, the goal of the cell is to survive long enough to reproduce. The cell accomplishes this by going through a journey called the cell cycle. The cell cycle has a strict order of stages, leading up to the big M. Step 1, the first gap. In gap 1, the cell has to go about its normal function, growing little by little. Step 2, synthesis. Once the cell is big enough, it enters into the synthesis stage, where it undergoes DNA replication. In this stage, the cell goes from being haploid to diploid, doubling its DNA. Next up, gap 2, where the cell grows even more and prepares to encounter the big M. All of these steps are part of interphase, and once it's complete, the big M, mitosis and/or meiosis. But first, the parts of the chromosome. A single section of the chromosome is called a chromatid. During DNA replication, each chromatid makes a copy of itself, and those two copies together are called sister chromatids. The part of the chromosome that links the two sections together is called a centromere. There are 5 main sections of mitosis: PMAT and cytokinesis. During prophase, the chromosomes come into view. That is, the DNA of the nucleus condenses into chromosomes, and the spindle microtubules unwind. During metaphase, the chromosomes line up on the metaphase plate in the middle of the cell, M stands for middle, and the spindle fibers attach. Next, in anaphase, the spindle fibers pull the chromosomes apart, and the two chromatids move to opposite sides of the cell. Then, in telophase, the DNA regroups and the cytoplasm starts to pinch. The nuclear envelope also becomes visible. After PMAT comes cytokinesis, where the cytoplasm splits the groups of DNA and organelles into 2 identical daughter cells. So that's mitosis. Now on to meiosis. Meiosis is a special process that results in special cells. Only germ cells undergo meiosis, and unlike mitosis, a cell can only go through this process once. The end result of meiosis is 4 genetically unique daughter cells, used for sexual reproduction and genetic recombination. Because we're producing more cells, this process is a little longer. It is broken up into 2 sections, meiosis 1 and meiosis 2, but each contains the same phases as mitosis, with slight alterations. During prophase of meiosis 1, the spindle microtubule unwinds and homologous chromosomes pair up and exchange DNA. With one maternal and one paternal chromosome, crossing over recombines the genes, making a unique set of DNA in the offspring. In metaphase, the homologous pairs line up on the metaphase plate with the spindle fibers attached. Anaphase pulls them apart, and telophase regroups the chromosomes into new nuclei. In cytokinesis, the cytoplasm is split into 2 cells, and meiosis 2 begins. After meiosis 1, the cell does not undergo DNA replication. Right now, each cell has a normal amount of chromosomes, but they are about to divide again. In prophase of meiosis 2, the spindle microtubules unwind. In metaphase, the chromosomes line up on each metaphase plate again, and the spindles attach. Then, in anaphase, the chromosomes are pulled apart to opposite sides of the cell. Telophase regroups the DNA until cytokinesis finally separates the cytoplasm, forming 4 unique daughter cells. In conclusion, mitosis creates somatic cells, is used for growth and repair, can be gone through multiple times, creates 2 identical daughter cells, and has 1 division. Whereas meiosis creates sex cells, is used for reproduction, can only be completed once, creates 4 unique daughter cells, and undergoes 2 divisions.